Thank you all for being here. My project for this, for the Laidlaw research, focused on the neuroscience of intellectual disabilities. So I'm just going to go straight into it. It's a big problem. The intellectual disability affects 2.5% of individuals. If you extrapolate that to the current world population, that's about 125,500,000 individuals. And it's not just the people who suffer from these intellectual disabilities, which is defined as an IQ of less than 70 points. It's also the families, friends, and everyone else that's around it. So it's a big problem. And what makes it even worse is that we have there is no known cure. There are no effective therapeutic methods to treat intellectual disabilities. And we can't fix something that we do not understand. So the point of my lab and my project was to really get at the molecular mechanisms, the reasoning behind these intellectual disabilities, and hopefully try to understand that to one day have the means to come up with effective therapeutic methods. So now, when I'm thinking about the brain, I like to actually think of playing Jenga. For those of you who have actually played Jenga, you know that it's comprised of many different parts, and there are some parts that you can move out and there's no problem, but then there are those very quintessential blocks that you cannot move, or the whole thing is going to come crashing down. And this helps me a lot thinking about the brain, because it's very resilient, but there are those things that you just cannot mess with, or the whole thing is going to come crashing down. And I'm going to keep coming back to this analogy. So first of all, I just want to set up for those who don't have a background in neuroscience, just understanding that the brain has a lot of neurons. That's about 86 billion neurons. And each one of those can synapse, can connect, communicate with about 10,000 others. And a synapse, basically that bridge between the neurons, trying to understand the flow of neurotransmitters from one end of the synapse to the other. And this is how the synapse is usually presented at, but actually this is a more accurate representation of it. And don't get caught up in all the names and everything that's going on here. I just want to emphasize the complexity of what's really going on in each individual neuron in the brain. And in this particular model, I want to focus on one key player, which is beta-catenin. This is one of those names that I'm going to have you sort of learn, but beta-catenin is a protein that you can see is both in the presynaptic portion and the postsynaptic portion, meaning in the neuron that's communicating and in the neuron it's talking to. And it's that blue figure in here. And you can see it's doing a lot of things. Beta-catenin is important for a lot of things involving communication between neurons, specifically holding that synapse together and sort of guiding information from the presynaptic component, just guiding information to where it needs to go. In a nutshell, it's important. And everything that beta-catenin does, it's important for a proper development of the developmental brain. So why do we care about this, and how does it connect to intellectual disabilities? Well, human genetic studies have discovered a very clear link between intellectual disabilities and a malfunction of the beta-catenin pathway. And even more so, sorry, if you can read it, it's just saying that. And even more so, individuals with disruptive mutation to the specific gene that codes for this protein, for beta-catenin, also have a high penetrance for intellectual disabilities. So what matters here is that intellectual disabilities is a big problem, and we think that this key player is the key to it. So that's what my project will focus on. So how do we go about studying this? Well, for the first portion of my research this first summer, we generated a conditional knockout mouse for beta-catenin, which in lay terms just means that we developed a mouse line that with the genetic techniques, we can delete, we can cut out the beta-catenin, and then just see what happens. And we do this in excitatory neurons in the forebrain, and we do it right after they are born, because if you do it before they're born, they actually die. That's how important it is. It has to be right after they're born, and in a period of major synaptogenesis and a period where the brain really begins to develop. So that was the first portion of the project. This is just to show this. Just focus on comparing the left column to the right, just how with the controls, the normal mice, they have normal levels of beta-catenin, and in the knockout, we just have a reduced number of beta-catenin. So this is just setting up what the mouse line is. And now we actually have to test how they perform. For those who are not familiar for this paradigm, this is called the Barnes maze task, which basically you put the mouse in the middle of an open space, and mice do not like being in open spaces, and they want to escape. There are all these possible routes you can escape through, and there's only one effective door out. And of course, at the beginning, they don't know it, but with trials and repetition, they eventually get to know it after like 3 or 4 trials for the controls. What's interesting here is with the knockouts, the ones that don't have beta-catenin, they don't learn. They just are always lost. They never learn to actually get out of it. And you can see here with the data that the latency to this goal, it goes down for the controls because they learn. But the knockouts, the ones that don't have beta-catenin, they don't get there. So there's an intellectual disability here. And this is just one of those tests that we use to sort of measure this. So just going back to this this image of the synapse, you can see that everything that beta-catenin touches, everything that the blue figure is touching, it's an important part. It's going back to the Jenga analogy. It's one of those pieces that you just cannot take out, or everything else is going to come crashing down. And one of those things is where we can measure that. It's not important to focus on the specific names, but P120 catenin, alpha N catenin, and cadherin, all these very important portions for the synaptic stability, they also are reduced in the knockout. Just compare the 2 different ones, the 2 different columns. The control, there's normal levels. And by the way, we're showing this through a Western blot. This is just to show the protein levels that are found in this tissue of the brain. And just comparing them, you can see that in the control, everything is fine, everything is normal. There can be proper learning happening. And in the knockout, everything is reduced. It's like that one Jenga piece that brings everything down. And the difference is significant. So just to wrap up this first point is that with the loss of beta-catenin, the loss of this protein in the knockout mouse, we see learning impairments and reduction of these synaptic binding proteins. So, and that was, then this work went on to being published in human molecular genetics, and I was very thankful to be part of it. So then I was, okay, can we conclude that just the more is better, more beta-catenin is good? It's not as easy in science. It's never quite as straightforward as more is better. So the second part of my research, the second summer, was just, okay, what happens when there's too much? Because now we show them when there's too little beta-catenin, there's learning impairments, there's loss of synaptic binding partners. And now let's try to look at a different version. Like what happens if we make a mouse line that has too much of a version of beta-catenin that, in a nutshell, it truncates, it splits. It's a different version that happens naturally in the brain, but when there's too much of it, we can also maybe see intellectual disabilities. So that's what we did. And to get to this point, I just need to introduce one more character in this whole play. This protein called APC, and it's a huge protein that also has a lot of roles in synaptic communication, and it has a relationship with beta-catenin, and it's huge, and it's going to become very evident why soon. So created a second mouse line, this one that now overexpresses beta-catenin, this specific version of beta-catenin, the truncated one. And just to see what happens, this is the beta-cat overexpressor. So the last mouse was a knockout. We deleted beta-catenin. In this one, we are overexpressing this version of beta-catenin. And this is just to show, again, what this looks like. In the beta-catenin overexpressor, there's a lot more of this truncated beta-cat. I'm not going to focus on this too much, but just I want to get to the learning impairments. Similarly to the knockout mouse, we see a reduction in the latency of the controls. They learn the task. They get to escape this room that they don't want to be in. After trials, after 3 or 4 trials, they get pretty good at it. But the overexpressor, they just don't get better. They don't get as better as quickly. And we even see in different tests that they have intellectual disabilities that are even worse than the knockout. And we believe that one of these processes is something called APC hyperphosphorylation. Don't get caught up with that big word. It only means that the phosphoryl sites are saturated. And when they're saturated, they cannot function properly. Okay? We see just in this figure that in the beta-catenin overexpressor, there seems to be something going on here. It's hyperphosphorylated, and we don't see that in the controls or in the other mouse lines. So we believe that maybe this has something to do with the observed intellectual disabilities that are going on. And even more interestingly is that we see an increased association of the beta-catenin, of this version of beta-catenin with APC, the player I mentioned before. And maybe this has something to do with it. Maybe just this attachment, the hyperphosphorylation, the presence of overexpression of beta-catenin is leading to intellectual disabilities. And this happens. And just a final point to really emphasize this, there is reduction of LTP, which in lay terms, it stands for long-term potentiation. And in lay terms, it's just how the neurons are able to fire together and wire together and are able to learn. And this is how the learning happens in the brain in normal subjects, and that is reduced as well in the overexpressor. So just to bring it back together, we saw in the first part that the loss of beta-catenin in the knockout mouse, again, learning impairments, loss of synaptic binding partners, and when there's too much of it, in the excessive one, in the overexpressor, there are also learning impairments. There's an increased association of this beta-catenin with APC and phosphorylation of APC, which is the saturation of this player. So just like in the Goldilocks zone, not too little, not too much. Going back to the Jenga tower, it has to have the right balance, not too little, not too much. And I just want to finally conclude by giving a shout out to this organization that actually focuses on the people with CTNNB1 syndrome, which is the gene that codes for beta-catenin. And just to sort of make the point to bring it back to the actual people, because I think it's easy to get like caught up in all the numbers and the proteins and the samples, but we have to remember that the actual people we're trying to help here, that there's real people with real suffering, and bring it back to that. So before questions, I just want to thanks to Jacob Lab, to Marie and to Sambeth, everyone in Jacob Lab for allowing me to be part of this process and being part of the research. So thank you very much. I can take questions.
Yeah, I'm curious about like I'm not sure you touched on it, but like what are maybe like cures on the market? What are they for this type of like learning impairment?
Yeah, I mean that's kind of the reason why this research is so important, because just getting at understanding the mechanisms behind it is how we can get close to like a cure, like a more traditional cure. But right now, to my knowledge, there is no cure. There are treatments, and there's this special training, special schooling, but there is nothing that can actually reverse a cognitive disability yet, which is why it is so important to understand the relationship of all these partners.
So I have two questions. So the first question is, I'm wondering if this study for this specific protein, if the study has happened also in other animal models, because from my understanding, sometimes mice can over-exaggerate or lie, actually.
Yeah, I mean, there's definitely like a caveat to using mice, and how confident can we be relating that to the human brain? There's a lot of overlapping and a lot of shared stuff between the mammalian species, which is why mice are good for a lot of these things. But you're right. I mean, for something like intellectual disabilities, like how can we test a mouse on whether or not they can do certain things, right? So we definitely have to take this data and the conclusions we can make from it with a grain of salt. But to my knowledge, I'm not sure if with other species we use like rats and mice, which are just for the ease of doing research with them and like the ethics. But I'm not sure if Marie. So yeah, so just like to answer the question, I don't know what it would look like with a different model. It's just that for the ease of the research, like trying like all the breathing and stuff like that has to do with it. We use mice and try to make conclusions from that. But I actually don't know with a different species. But it's a good question. Yeah.
How do you generate those beta-catenin in knockout mice?
Yeah. So I mean, the actual technique is like kind of like the nitty gritty of it, but basically we use like Cre recombinase, which you can basically inject something in the DNA through breathing, and then you present a certain catalyst. It's like the Cre recombinase sets up the specific sites of the gene that you want to cut out, and then you can present the actual Cre, and it will cut at those sites that you basically bred them for. So it's like they're just like genetic techniques. You can be very specific and be like, okay, we want to cut just the beta-catenin gene, and we can set up sort of these cutting sites, and then we present the Cre, and then it cuts out, and then through breeding, we have a whole line of mice that don't have it. But it's very technical with the actual genetic techniques, and with be careful with the breeding, because then if they mix, then it's kind of hard to tell, and you have to do Western blots to make sure, and like genotyping, as you know, is that just to make sure that every mice we're testing is either a control or a knockout, and we're certain of it. But that's basically how it goes. I can actually go into the more detail later, but in a nutshell, that's how it works. More questions?
I've got one. In the human population that you were talking about, obviously not all 2.5% are beta carotene intellectual disabilities. Do you have a number that is could be attributed to that?
I'm not sure. I mean, the actual people that have this specific syndrome are not a lot, just because it's a very specific kind of syndrome, and a specific, like, to actually be diagnosed with it, it's kind of hard. So this specific organization, for example, just have, I mean, I think at the end of this, they say that in the world, there were like 200 and something people with this specific syndrome, but it's kind of hard to discern, just because they put intellectual disabilities all under one bucket. And they just get that number of 2.5%. But I don't know off the top of my head the specific number of beta-catenin, but for example, this organization gets to a number of around like 250, 300 people in the world that have been diagnosed with just the gene coding for beta-catenin. But there's probably so much like comorbidity of things going on at the same time that it's kind of hard to discern yet how specific it is. But that's all I know for now.
Okay, thank you.
Thank you.